The overall goal of this two-dimensional semi-denaturing detergent agarose gel electrophoresis is to confirm size heterogeneity of amyloid or amyloid-like fibers seen during traditional SDD-AGE.
This method can help answer key questions in the amyloid or protein aggregation fields, such as whether size heterogeneity seen during traditional SDD-AGE is the native state of the fibers in vivo or the result of protein degradation or dissociation during gel electrophoresis. The main advantage of this technique is it can be performed easily in lab without specialized equipment. No additional equipment is required beyond what's used in traditional SDD-AGE.
First, seed the amyloid-producing HT29 colon cancer cells in a 10 cm tissue culture dish. Then, incubate the cells overnight at 37 degrees Celsius with 5% carbon dioxide. Once the cells reach 80% confluency, use phosphate-buffered saline to wash the cells. Then, add 3 milliliters of trypsin to the cells and incubate at 37 degrees Celsius for 3 minutes. After the cells have detached from the culture dish, add 10 milliliters of culture medium to the plate. Then transfer the cell suspension to a 15 milliliter conical tube. Next, centrifuge the cell suspension at 1000 times G for 3 minutes at room temperature. After the centrifugation, aspirate the medium and resuspend the cell pellet in 5 milliliters of culture medium. Count the cells using a